Hello friends, today we are going to study about protein sequencing. First of all, can we give the answer of the question how proteins are so different chemically, functionally and structurally? Proteins are very diverse, chemically also, functionally also and structurally also. So what is the reason behind their diversity? The reason is the primary structure of the protein. Now what we mean by primary structure? Primary structure is nothing but a sequence of amino acids present in a protein and their composition. So what all amino acids present in a protein constitutes its primary structure, which has two parts: the different types of amino acid it is made up of and the sequence of those amino acids. But compared to these two, the sequence of amino acid is more important. Now, we all know that the proteins have an unique 3D structure. Because of their unique 3D structure, they have their unique function. We also know that proteins has unique amino acid sequence. So can we say that these amino acid sequences are only responsible for their unique function or their 3D structure? Yes, we can say. But what all observational evidence we have for that? The first, proteins with different functions always have different amino acid sequences. This is the first observation that proves that there is a relation between amino acid sequence and the function of the particular protein. Because the proteins with different function always have different amino acid sequences. Second observation, similar proteins, the proteins that are functionally similar even from different species, often have similar amino acid sequences. This also suggests that there is a relation between the amino acid sequence and the function of a protein, because if the protein function is similar, the amino acid sequences are also similar even from different species. Third observation is human genetic diseases are due to defective proteins. There are many human genetic diseases which are due to defective proteins. And from those diseases, 1/3 is because of change in their amino acid sequences. So this also suggests that amino acid sequence has an importance for the protein function and the structure. So now, can we say that the amino acid sequences are totally fixed? No. The amino acid sequences are not absolutely fixed. Flexibility is allowed. In case of humans, 20 to 30% of proteins are polymorphic in nature. That means having amino acid sequence variant. So, some flexibility is allowed. It is not like that amino acid sequence should be absolutely fixed. Therefore, what we can say, the amino acid sequence in some regions of the primary structure might vary without affecting the biological function. In the whole protein, if we have this long amino acid sequence, say for example this particular middle sequence, if this sequence vary, the function will vary. But if this sequence vary, the function will not vary. So that means in amino acid sequences, there are regions in the primary structure which can vary without affecting the biological function. And also, there are some crucial regions which are essential to the function of the protein. And if those sequences get vary, then the function will affect. So those sequences are therefore conserved in nature. So for a protein, the whole amino acid sequence or the whole primary structure is not important. There are regions where variations are allowed without affecting the biological function, and there are regions where variations will cause defective proteins. So those sequences should be conserved. Now, first we'll study about the sequencing of short polypeptide. How we will sequence a short polypeptide. The step 1 is, first we have to determine the N-terminal amino acid residues. If a polypeptide is present, it will obviously have one C-terminal end and one N-terminal end. So we have to determine the amino acid which is present in this N-terminal. This is the first step of sequencing for any short polypeptide. Now how we will determine the N-terminal residues? So for that we can use various chemicals. We can use densyl chloride. We can use dapsyl chloride. We can use 1,fluoro-2,4-dinitrobenzene. In this example, this chemical is used, but it is easy to determine the N-terminal with the help of densyl and dapsyl chloride because they fluoresce. So in case of 1,fluoro-2,4-dinitrobenzene, when it is added to the polypeptide, it gets attached with the N-terminal amino acid, forming 2,4-dinitrophenyl derivative of polypeptide. In an acidic condition, this is HCl. So in an acidic condition, what happens? The peptide bonds are broken. A polypeptide chain are made up of, for example, these 4 amino acids. So in the acidic condition, all the peptide bond will be broken. So all the amino acid will get released. Now this is the N-terminal residue. With this, a chemical is attached. This is this part, 2,4-dinitrophenyl derivative of amino terminal residue plus free amino acids. These are free amino acid. This is the N-terminal amino acid residue with which the chemical is attached. In this case, the chemical used is 1,fluoro-2,4-dinitrobenzene, but we can also use densyl or dapsyl chloride. So this amino acid will then determined or identified. So this is the first step of sequencing of short polypeptide, that we have to identify the N-terminal amino acid residues. What is the importance of this step? With the help of this step, we can able to know whether the protein consists of more than one polypeptide chain. If suppose a protein has 2 polypeptide chain, so this will also have an N-terminal, C-terminal. This chain will also have an N-terminal, C-terminal. So by determining the amino acids present in the N-terminal, we can say whether the protein is made up of one or more than one polypeptide chains. The next step is we have to sequence by Edmond degradation. How we can sequence through Edmond degradation? The Edmond chemical is phenyl isothiocyanate. This chemical, an alkaline condition, they'll form PTC adduct. What is PTC? Phenyl thiocarbamyl. So they'll form a phenyl thiocarbamyl adduct with the amino acid present on the N-terminal. Then in the presence of trifluoroacetic acid, they will form one derivative, which is in the presence of acidic condition will convert into a more stable derivative. So again, we have a polypeptide consisting of, suppose, these 4 amino acids. We'll add this phenyl isothiocyanate. It will come and react with this amino acid. Then when we will treat with trifluoroacetic acid, it will break this particular bond. So after breakage of this bond, the rest of the polypeptide chain is intact. Only the amino acid present towards N-terminal is with the added chemical. This amino acid is then determined, and rest of this polypeptide or peptide will again return to the same reaction. So in the second step, what will happen? This amino acid will get separated and identified. In the third step, this amino acid will get separated and identified, and then this amino acid will get identified. So like this, one by one, every amino acid is getting identified through the process of Edmond degradation. So this how we can sequence the whole polypeptide, because we'll come to know about the amino acid present. Say for example, this is glycine. Second time, in the second cycle, we found that this amino acid is proline. Next, in the second cycle, say for example, we found this amino acid is tyrosine. So like that, we can easily then sequence a short polypeptide by Edmond degradation method. Now, the efficiency of the method. The efficiency of the method depends upon the efficiency of the chemical step. Now, in case of Edmond degradation, let's understand the efficiency with an example. Suppose a peptide molecule consists of 4 amino acids. Say for example, glycine. This is N-terminal. This is C-terminal. So glycine, proline, tyrosine, tryptophan, for example. Now, the chemical is 97% efficient. Now in a solution, 100 molecules of this peptide is present, and we have to sequence the peptide. The efficiency is 97%. Now, first the chemical will bind to this glycine. It will get separated and identified. Because the chemical is 97% efficient, so from 100 molecules, from 97 molecules, glycine will be separated out. But 3 molecules of this peptide is still in the solution that has glycine attached with it, because the chemical is 97% accurate. This is the first round or the first cycle. From 100 molecules, from 97 molecules, glycine has been identified and separated out. But 3 molecules of the peptide are still there in the solution that contain glycine, still in their N-terminal. Now the second round starts. From this 97 molecule, the chemical is 97% efficient. So from this step, again, 97% of 97. How much it will be? 94.09. Left is 2.91. Because from this molecule, glycine has been identified and removed, the next amino acid is proline. So it is efficient 97%. So this is proline will be identified and removed. But 2.91 will still have proline. Coming to this 97% accurate. So 2.91 and the remaining is 0.09. So from 2.91, glycine will be identified and removed, and in 0.09, glycine is still present. So like this, it will go on. After second round, if we see what we can say, this we can consider as a round figure 3 molecules, and this is also we can consider it as 3 molecule, and this we can consider it as 0.1. 0.09 is 0.1. So that means every time with each cycles, because the step is 97% accurate, so this background noise or the error will go on increasing. So the efficiency of the method depends upon the individual chemical steps. This Edmond degradation method is now automated using a machine called sequenator. In this automatic machine, the chemicals are added appropriately, separated, amino acid is identified, and data is recorded. And this automated machine is more than 99% accurate. So nowadays, this automatic machine is used for sequencing of short polypeptides because the machine is more than 99% accurate. So this background noise will be less. Otherwise, it will create problem and we cannot tell the exact sequence of amino acid, because every time when we are getting the exact amino acids, we are also getting the other amino acids. So they will confuse the result. They'll disturb our result and we cannot land up with an accurate sequence of amino acid. So the method is automated, and this automated method is more than 99% accurate. The first step is we have to identify the N-terminal residues, because to know how many polypeptides a protein is made up of. And the second step is we have to sequence those polypeptides using Edmond degradation method. Now one question from my end for you all, that is, can we determine the number of polypeptides in a protein if the polypeptides have same N-terminal amino acid residues? What we learned is we have to determine the N-terminal residues because to know the number of polypeptides, because all the polypeptides will have their N-terminal and C-terminal. So how we can know that a protein is made up of 3 polypeptides? For example, when we are performing the first step, say for example, we got glycine identified, we got proline identified, we got tyrosine identified. So we got 3 different amino acids identified in the first step. So we can say that the protein is made up of 3 polypeptides. Now, if suppose all these polypeptides have glycine in their N-terminal, then can we identify the number of polypeptides that particular protein constitute of? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.